Today we will discuss the structure of viruses. First of all, definition of viruses, that how we can define virus. Viruses are microscopic organisms that can infect hosts like human, plants, or animals. They are a small piece of genetic information, DNA or RNA, inside a protective shell made up of protein called capsid. Some viruses also have an extra envelope around the capsid. Viruses can't reproduce without a host. So this is a simple definition of virus. Now, introduction. Viruses are smaller and simple in structure than unicellular microorganisms like bacteria, protozoa, fungi, chlamydia, etc. Viruses contain only one type of nucleic acid, either DNA or RNA, but never both. As viruses have no ribosomes, mitochondria, or any other cellular organelles for energy production and protein synthesis, they are completely dependent on their cellular hosts. They replicate only within a cell of the host that they infect. Unlike microorganisms, many viruses can reproduce themselves from their genome, but only inside the suitable host, suitable cell. A single nucleic acid molecule, that is, nucleic acid of a virus, alone is infectious. Outside a susceptible cell, virus particle is metabolically inert. Now, what is susceptible cell? Susceptible cell means a cell in which virus particle can replicate. So, virus particle is metabolically inactive when it is not present in the cell. On the other hand, when replicating in a cell, it exhibits all the characteristics of life. Studies have shown that virus particles or virions range from about the size of smallest unicellular microorganism, that is 300 nanometer, to objects little bigger than the largest protein molecule, that is 20 nanometer. It means that size of the virus particle or virion ranges from 20 nanometer to 300 nanometer. In the simpler viruses or virion consists of one single molecule of nucleic acid surrounded by a protein coat called capsid. When capsid and its enclosed nucleic acid present together, it forms nucleocapsid. Students generally ask a question that are viruses alive? Now, outside a susceptible cell, virus particle, just like bacterial spore, is totally metabolically inactive or inert. But on the other hand, when replicating in a cell, when present in a cell, it exhibits all the characteristics of life. It means that outside a susceptible cell, virus particle is inactive. It is active only when it is present inside the susceptible cell. Now I'm using word virion again and again and virus. Now what is the difference between these two terms, virus and virion? Main difference between virus and virion is that virus is the nucleoprotein particle, whereas virion is the active infectious form of virus. It means that virus particle outside a cell, it is inactive, right? When it enters a cell and become active, replicating, we use the term virus particle or virion. A virus consists of genetic material and it's covered by a capsid made up of protein, but it doesn't show any metabolic activity when not present in the cell. We use the term virus for that, right? Virion is the vector stage of virus. Now, what is vector? Vector can deliver the materials by infecting the cell. Vector stage means a disease-causing stage that can transmit disease, allows the transmission of virus from an infected host cell to another host cell. Therefore, the virus is the extracellular phase, whereas virion is the intracellular phase of virus. Morphology of virus. For many years, it has been known that viruses are smaller than unicellular microorganisms, but it became clear from the studies of various scientists as in 1898 to 1899, Dutch plant pathologist Wizernik working on tobacco mosaic disease. Similarly, the German veterinarians Loeffler and Froch, they were working on foot and mouth disease of cattle. Tobacco mosaic disease and foot and mouth disease of cattle both are viral diseases. Results of the studies showed that these diseases could be transmitted by material which could pass through a filter with pores which are too small to allow passage of bacteria. So this new group of microorganisms became known then as a filtrable viruses. You can see in this diagram tobacco mosaic virus when attack or infect a plant, symptoms appear like this. Symptoms on the leaves, they appear like this. And tobacco mosaic virus, it looks like this. So here, central core is there, surrounded by this genetic material, that is RNA. And this genetic material, again surrounded by protein molecule, a layer that is capsid made up of protein. And that capsid again made up of small morphological units called capsomeres. So this view means in cross section. When in means in surface view, this tobacco mosaic virus, it looks like this. For a time, they were also called ultra microscopic. Why? Because they are too small to be seen with ordinary light microscope. Only with the advent of electron microscope, it became possible to study the morphology of virus in detail. In 1959, knowledge of viral ultrastructure then was transformed when Brenner and Horney applied negative staining to electron microscopy of viruses. Because potassium phosphotungstate, which is electron dense stain, it, fill, it fills the interstices of the viral surface and it giving the resulting electron micrograph means a degree of detailed which was not previously possible to study. Now we will study about the virus structure. This in this diagram you can see basic virus structure. As we were talking about that only two components are present in a virus structure. Protein coat, that is capsid, it surrounds the genetic material of virus. So when both are present, it forms a complete structure that is nucleocapsid, means capsid plus nucleic acid, right? In the simpler viruses or virion, it consists of two components, a single molecule of nucleic acid surrounded by a protein coat called capsid. So when capsid and enclosed nucleic acid present together, it forms nucleocapsid. But in some complex viruses, capsid surrounds a protein core. And in other viruses, this capsid is surrounded by an envelope made up of lipid and protein. The capsid is composed of morphological units called capsomers. Capsid, which is made up of protein, again made up of morphological units. Those units are known as capsomeres. And those capsomeres were held together by non-covalent bonds. Individual capsomeres, which consist of one or more polypeptide molecules, are visible by electron microscope only. Now, capsid made up of protein. Each capsid is made up of identical morphological units, capsomeres, and each capsomeres made up of polypeptide molecule. Now, what is polypeptide molecule? Polypeptide is an unbranched chain of amino acids that are linked together by polypeptide bonds, right? So in helical nucleocapsid, the viral nucleic acid is folded throughout its length in a specific relationship with the capsomers, right? Here you can see again example of tobacco mosaic virus. These are the capsomere. Capsomers, capsid surround a protein core, that is RNA, and this capsid is made up of capsomeres. You can see in this diagram, all these are capsomeres. Here in cross section, same structure. So this is nucleocapsid, means nucleic acid and capsids, and each capsid, that is outer layer, this is capsid, so it is made up of capsomers. Within an infected cell, the capsomers of the simpler viruses, they self-assemble to form capsid. The manner of their assembly, means how the capsomers assembled, is strictly defined by the nature of bonds formed between individual capsomers. Now, this nature of the bonds that is formed between the capsomers, this imparts symmetry to the capsid. How many kinds of symmetry are there in the viruses? How this symmetry is formed, we will discuss in detail in the next lecture, but a little idea I will give you here. This is bacteriophage and this is coronavirus. These two type of the viruses are there. The coronavirus symmetry is helical, means it forms helix, just like in the case of tobacco mosaic virus. But in bacteriophage, you can see this is the head of bacteriophage and this is a tail part. So head, symmetry of the head is icosahedral. So how these two symmetries are formed, we will discuss in the next lecture. Now, what type of the question, whatever we have discussed, what type of the question can be asked in UPSC? Like difference between virus and virion, or discuss virus structure in detail, means general structure of virus. So these type of the question generally can be asked in the UPSC exam. So in the next lecture, we will discuss symmetry and chemical composition of viruses in detail.